Sir, you can start, sir.
Yes, ma'am. Okay, thank you, ma'am.
Good evening, sir.
Good evening, Jaswant.
Good evening, sir.
Good evening, Ajay. Okay. Shall we start now? Okay.
Yes, sir.
Okay. See you. Last class, what we have discussed, will you please? What are the things already we discussed? Any idea, my dears? Yes. Today we are going to discuss about the last part of the reproductive health. Okay. So already we discussed what are the reproductive health, particularly. See that significance of reproductive health and reproductive health problems and strategies. And what are the strategies is known: early marriage, personal hygienic, population explosion. Okay. And also sex determination by the amniocentesis. And then amniocentesis is nothing but one of the technique to identify the and also check at the sexes, whether the baby is female or male. And not only that, and also we may check fetal infection, chromosomal genetic defects, and abnormalities. Okay. So this is another one. And also population explosion, birth control already we discussed. In note it down, this is very, very important. Which one? Which year the population explosion is happened? 1951. Don't forget it. 1951. Hello. Okay. Next, what are the reason for the population explosion? Already we discussed mortality, birth rate, and there are other facilities, health increased facilities also, but even the living conditions, that things also already we discussed. This is very, very important. Don't forget it. The correct answer is 151. 1951 is the correct answer for the population explosion first occurred. Second, 1981, like that you may note it down. This is very, very important. Okay, and birth control, kindly go to the refer the books. Already told you. And then types of intrauterine devices. Okay, already we saw it already. These things also. And hormonal method, that things also already we discussed. And combined oral pills, the Mala D, Mala N. How many days we have to take? All those things already we discussed. It depends upon the mixture of hormones. What are the hormones? You know, progesterone and also called as progestin, along with the estrogen. We have to provide 21 days. That things also already we discussed. And what are the essential components? Only pituitary hormone they can secrete, FSH and LH. This is also very, very important one. And then we'll pass on to the next one is mini pills. Already we discussed progesterone only pills, POP, simply called as how many days we have to take. All those things already we discussed in the previous classes. So centochromum, man, this is also another one. It is also an anti-estrogenic effect. Anti-estrogenic effect done by which one, you know? The centochromum. So this is another type. This is very, very important. Central Drug Research Institute, where it located already in our Lucknow, Madhya Pradesh, and it has been released in India under the name is called as Sahili. Okay, note it down. This is very, very important one. The Central Drug Research Institute in our Lucknow. So many times they were asked the question that match the following aspects. Other sometimes it may be direct questions also there. they were asked. Okay, no need to forget. So depot injection also already we discussed. This is also collectively called as what is the DPMA, otherwise called as Depo-Provera. How many months we have to provide? 3 months. This is also called as missing pills. Already we discussed. Suppose when you are missing this pills, you have to take the injections. Next, we are going to discuss about the important part, that is NAR plant. What is by NAR plant? See here, NAR plant contains 6 small permeable capsules. How many capsules present in it? 6 small permeable capsules. The depth and width also given is 34 millimeter into 2.4 millimeter, each having about 36 milligram of levonorgestrel. Note it down. This is the composition. The NAR plant contain the component is called as levonorgestrel, made up of silicosane material. Note it down. These are the 2 important things. Very, very important. Silosane material. Silosane material. Note it down. This is very, very important. NAR plant. It is a small 6 capsules. And also how many milligrams? 6 into 6. See here, 60, 36 milligram levonorgestrel, made up of silosane material, and each containing progesterone or levonorgestrel. It is effect lasts for 5 years. The implant suppress ovulation by main action is suppressing endometrium. What is the main functions of NAR plant? It is the implant suppresses of ovulations. This is very, very important. Let me discuss so many times. What is the main functions of the implantation? Suppose if it is pregnancy, is nothing but the implantation of the ovulation on the endometrium, isn't it? This is very, very important one. It prevented by which one? One of the chemical substances that is called as NAR plant. It contains 6 small capsules, 36 mg of levonorgestrel, made up of silosane, silosane, okay, materials. Okay, this is very, very important. What is the main mechanism of this one? Implant suppress ovulation by the action of suppressing endometrium. So go to the next one. It takes 5 to 10 minutes to insert under local anesthesia. Sometimes they were used for the local anesthesia. Anesthesia is what is that? Mostly used for the operation process. Okay. Okay. Open heart surgery, whatever it may be. Local anesthesia means it's a very mild dosage. Okay. Signs the capsules are not degradable. They need removal at the end of its use or earlier if the women wish to stop the use of contraception. Okay, so their own interest they can remove. That is very, very important. They can easily stop the usage of contraceptions. These are the very, very important things. Okay. Okay. So go to the next one. Say the insertion and removal is made easier by using a single rod implant, and its effect lasts for 3 years. Okay, this is the implant. So single rod is also used for the implant. Okay, surgical sterilization. Already we discussed so many sterilization methodology is present in it. Particularly, there are 2 types of sterilization. Sterilization, nothing but what is that? There is a small operation, permanently remove or simply tied the cut and tied. That is the process is called a sterilization. See here, the sterilization operation is undertaken with the primary objective of preventing further pregnancy permanently. Okay, so sterilization nothing but a small operation. Okay, what will happen? It completely prevent further pregnancy. They will not get any child after the sterilization process. Here, this not depends upon the temperature when you are going to sterilize the foodstuffs or any other milk or something else like that. It's not like that. It is also type. What is that? It's nothing but the operation. Sterilization is nothing but one of the process. But here they are taking surgical sterilization. Means completely removal or cut and tie the particular passage. Permanently, we will not get the child. There are two types of is there. One is called as vasectomy, tubectomy. Vasectomy mostly where it will take, whether male or female reproductive organs. Tubectomy where? So tubectomy is mostly in the female. Vasectomy is nothing but the male. Okay, we will see one by one deeply. Okay, see here. So vasectomy, the sterilization procedure in the male vasectomy is nothing but the male is called as vasectomy, in which a small part of the vas differences is removed or tied up through a small incision on the scrotum. This is a process. Vasectomy means you have to identify which part they were used, they were removed or tied, like that. You have to note it down. This is called as vas differences. Vasectomy, vas differences is removed or tied after cut, and they have to make a small what is that, knot like structures. So as a result, what happened is they will not produce their sperm, they will not pass through the way of sperm. Okay, this is the important thing. Once again, repeat me. Vasectomy, male. Which part? Was difference is removed or tied up. Okay, see here. So this is a structure, is a male already in our vasectomy. This is the normal one. So this is the structure of vasectomy. Epididymis, sperm, storage of sac, testicle. So already this is the male reproductive part, particularly this part completely cut. See here in this portion, B part, they cut and also tied. As a result, what happened? They will not produce their sperm. They will not move the sperm from the which one, the testicle to the which one, epididymis, and also these two vas differences. This is the part is called as, what is that? Sterilization, male sterilization, otherwise called as vasectomy. So this is another part. This is the male one. See here, this is the part. Here there is a cut. Okay, this is also vasectomy surgery. Okay, so this is also another one. See here. So this is a part. They made a cut, and also what happened? Already tied itself. As a result, what happened? They will not produce, but they cannot move through the way. Okay, so okay. Next one is called as tubectomy. Tubectomy is nothing but one of the sterilization procedure in the female is called as tubectomy. Sorry, here the spelling mistake is there. In which a small part of the fallopian tube, note it down this point, there is vas differences. Here is the fallopian tube is removed or tied up through a small incision in the abdomen of the through copulatory organ, otherwise called as, there is a name, copulatory organ. So here the which one, fallopian tube, don't forget it. There was differences. That's one tube. Here tubectomy. Was differences, vasectomy. Note it down. Each one it has a specific what is a respective name also. Okay, according to their removal part. This is the female reproductive organ already on. This is a normal uterus. Sir, where is the ligaments, the infundibulums, ligaments, fundus, and fembria? So all this is the normal one. Okay, see the next one. So this is called as tubectomy. Fallopian tube is here. Okay, here they made a cut here. Okay, and also tied with the knot. As a result, what happened? The production of ovum or egg, and also captured by the which one, the fimbriae, and also move on to this part. This is called as a infundibulum. Okay, all those things already know. Fallopian tubes. Suppose if it is reached the fallopian tube, there is a what is a knot is there, so they cannot allow to the uterus. So as a result, what happened? What happened? The child is that means the pregnancy is not possible. Okay, see the next one is also same. See that sperm is also moved this way, but it do not reach the egg because here they made the tie, cut and tie. Okay, the process also done by the doctors. So this is called as tubectomy. There is fallopian tubes is also cut. There is no connection between these. Already you know about that thing. So once it reaches the egg also, it will not move this up to this level only they can move. Further, it will not move to the uterus. So the sperm is waiting here. It will not join the egg, and also there is no possibility to form a zygote. Okay, note it down. Don't forget. Sometimes they may ask the question, a diagrammatical question. Don't forget it. Don't forget it. Okay, so okay, next one is a very, very important one. Sexually transmitted diseases. There are so many sexually transmitted diseases also reported by the many scientists. AIDS is also the syphilis and conchoid, denovasius, lymphogranuloma, vernoma, gonorrhea, non-gonococcus genital infection. These are the so many things. Even taken examples of HIV. Okay, these are all comes under the sexually transmitted only through the sexual contact only. Okay, syphilis. Here you must note it down. First thing, which type of disease? Okay, which disease? First of all, which disease? Which one of the cosmetic organism? You must note it down. They're very, very one. And then symptoms. Already I told you symptoms. And fourth one is called as what is a diagnostic process. Okay, so many times they were asked the question syphilis and gonorrhea. Syphilis, the cosmetic organism is Trypanosoma pallidum. Note it down, my dear students. Take a note. Syphilis is one of the sexually transmitted disease caused by the cosmetic organism is called as Trypanosoma pallidum. Trypanosoma pallidum, whatever it may be. So this is very, very important. And second one is called as gonorrhea. Yes, gonorrhea. Okay, so this is also very, very important one. Okay, see here, gonorrhea is caused by which one? You know, Neisseria gonorrhea. Note it down. This is very, very important. So many times they were asked the question. Just to go and check it in any NEET question. These two question will be there. Okay, just don't forget it. Syphilis, Trypanosoma pallidum, and gonorrhea, Neisseria gonorrhea. Okay, these are all some of them are bacterial diseases, some of them are viral diseases, some of them are fungal diseases also there. Okay, that things we are going to discuss one by one. Chancroid, Haemophilus, see here, decree. Okay, Denovanicis and Calamantobacterium granulomatis, Lymphogranuloma venereum. Okay, Calamida trachomatis, gonorrhea. Here you must note it down. These two only, syphilis and gonorrhea. Okay, Neisseria gonorrhea. Neisseria gonorrhea. Sorry, Neisseria gonorrhea. Non-gonococcus genital infection. Genital infection. Calamida trichomatis. Uroplasma ureolyticum. Okay, another one is called as what is that? Womanus vaginalis. These are the things. See here. These are the types of microorganisms mostly present in our body during the sexual contact. Okay, human immunodeficiency virus. Already know this is very, very and harmful and effective virus. Okay, worldwide problem already in our but okay. So this was introduced in the year 1981 and also 1984. The India were discovered this type of viruses. Coronavirus already you know, but there are so many traditional. Okay, so coronavirus is also already discussed. So many what is a mutated virus also there. Okay, and varicella, Zoster virus, hepatovirus A. These are all are viruses, isn't it? Here, bacteriophage is also such. This type of bacteriophage is also Staphylococcus bacteria, meningococcus. What is that bacteria type? It is also having a flagella-like structures. Pneumococcus, see here, and also colon bacillus, Helicobacter, and also another one is called as what is that? See here, this is also bacillus rod type of bacteria, and Salmonella. These are the what is that? cosmetic organism. Okay, see that chlamydia, the structure will be look like this one. Gonorrhea, already told you, this is also a another type. Mycoplasma, already we discussed, there is no cell wall, lack of cell wall, anaerobic condition, they can easily alive in nature, but they only produce the disease only. Mycoplasma, ureaplasma, note it down, these are the another. See, the next one is called as major sexual transmitted diseases we are going to see. Here, see that already discussed, virus is the bacteria, fungi, protozoa, and insects. These are all are the cosmetic agents. And a specific name, which organs, which virus is caused. What are the diseases here? Virus, first one, take an example. Human papilloma virus. The disease called as RODS. Second one is herpes simplex, herpes. Hepatitis B, hepatitis, HIV, AIDS. These are all are virus causing diseases. The name of the agent or microorganisms. What about bacteria? Chlamydia. Neisseria gonorrhea is one of the bacterial diseases. Sometimes they may ask the question, how you know? The Neisseria gonorrhea is a bacterial disease, viral disease, or fungi disease, or protozoa disease, like that they ask. Gonorrhea is caused by the cosmetic organisms, like that they may ask the questions. Okay, see here, Trypanomia pallidum, syphilis. Syphilis caused by the cosmetic organism Trypanomia pallidum. Syphilis is a bacterial disease, viral disease, fungi disease, protozoa disease, like that they may ask the question. These are the two very, very important one. Hemophilus ducrei, conchroid, already we discussed. Fungi, already you know, fungi is one of the achlorophyll nature, mostly found in the moisturizing habitat. Okay, the Candida albicans, candidiasis, note it down. This is very, very sexually transmitted disease also. Which one of the microorganisms? Fungi is also participate. Fungi is also involved in these diseases. The name is called as Candida albicans, fungal disease, candidiasis, and protozoa, Trichomonas vaginalis, trichomoniasis. This is also another type, female. Insects, see, scabies. Okay, scabies is also a disease caused by the insect. What is the name? I couldn't see here. Strobili, like that this comes. Sarco, like that this comes. Okay, I couldn't visit. Okay, these are the major sexually transmitted diseases also that. So you have to careful. Yes, Sarcoptes scapi. Sarcoptes scapi. Okay, the scabies is there. Okay, go to the next one. So bacteria, just can you see this slide, my dear students? Ajay, can you
Yes,
see
sir.
this?
Yes,
Yes, just okay. So first one is called as a type of organism. Bacteria has given here 4. First one is gonorrhea, chlamydiasis, choroid, soft. What is it? Chinery, syphilis. Okay, Neisseria gonorrhea, already told you. And also chlamydia and trichomatis, homophilus and ducreis, and Trypanomia pallidum. Okay, see, first one by one we will see. Bacteria, gonorrhea, Neisseria gonorrhea is a cosmetic organism. See what are the symptoms is there? Inflammation of the urinogenital tract. We cannot be able to urine properly. So when you are urination, the irritation is also there. So the patient feels a pain or burning sensation during urinations. Okay, this is a very, very complicated due to the presence of the microorganism Neisseria gonorrhea. This is also cosmetic organism, sexually transmitted gonorrhea. Discharge of pus also during the urination. Discharge of pus also is eliminated during the urination. How to detect gram staining of a discharge culture? When you are going to culture, okay, we can easily identify these type of diseases. The culture, blood culture is there. So many culture is there. Here the gram staining of discharge culture, the urine samples also taken, and also under the culture technique, there were culture for 1 or 2 days, you will get the sample. After that, the collected samples also examined under the microscope with staining. Okay, gram staining. There are 2 types of staining. Already know everybody, isn't it? Okay, bacteria. So another one type is chlamydiasis. Chlamydiasis trichomatis. Woman, cause of inflammation of cervix, urethra, and urinary tubes. Similar to the it may transfer into the pelvic inflammatory disease of PID, causing sterility. Some of the women, they cannot be able to produce their child, means due to this disease also. This bacteria also is there inside. Clinical gram staining of the discharge, antigen detection. These are the 2 are same. This is male, this is also a female. But men, the characterized by a thick pus like a discharge and painful urination in male. Always sexually transmitted, sexual organ will be affected. That is very, very important due to the microorganism. That is very, very important. Okay, bacteria, Chancroid, soft and Chancery. Haemophilus ducreyi, the painful dirty ulcers localized anywhere on the external genitalia, outer part of the urinal part, they are having some ulcers. There is a wound-like structure is there. Development of painful enlarged lymph node in the groin. The lower part, there is enlargements also there for scrotum or whatever it may be, like that. Bacteria. Syphilis, already told you. Trypanosoma pallidum occurs in 3 stages. Note it down. This is very, very important. Trypanosoma pallidum is also it has 3 stages affected. Primary syphilis, secondary syphilis, and tertiary syphilis. Primary syphilis, hard, dry, and also bacteria-filled sore called chancre, otherwise called as, appear on the genitalia. The outer part will be, there is a small hard and bacteria-filled sore. Okay, so here the clinical culture. Here the clinical culture. Same, all are clinical culture only. Secondary, what disseminated syphilis. Several months later, multiple lesions on the mucous membrane on the lips, mouth, and genitalia, and other symptoms include the white patches on the tongue, cheek, and gums, low-grade fever. These are the second symptom. Okay, lips, mouth, and genital. Other symptoms included, while the patches, small dots like this, wound-like structures present the every part, lips, mouth, and so on. So, tertiary syphilis is there. Okay, see this is another one. Bacterial infections, organs cannot be visible. This one also. Yes, it is characterized by the spread of bacteria to internal organs like heart and brain, and maybe lead to the paralysis, mental disorder. Bacteria may be crash even the placenta of an infected mother to the newborn during birth. These are all identified with the detection technique is called as the Wassmann test. Note it down, my dear student. Trypanosoma pallidum is also detected by the technique is called as Wassmann test or Wassmann reaction. Is an antibody test for syphilis. And also last one is called as ELISA. Sometimes they may use for the ELISA test for the identification of Trypanosoma pallidum. So this is very, very important. What are the things already? All are bacterial diseases only: gonorrhea, chlamydiasis, and Chancroid, syphilis. You have to note it down. Cause of the organism. What are the symptoms? Detecting techniques. These are the very, very important. Particularly gonorrhea and syphilis is very, very important. Without these questions, there is no neat question paper. Just go through it once again and go to the next one. Is see that these are the types, these are the different types of bacteria and viruses also present in our body. Okay, during the sexually transmitted. Okay, so we cannot be able to each one know it. These are the types. You just note it down. How many types of bacteria, virus, and different types of causative organisms also involved in the which one sexually transmitted diseases. So the bacteria, already we discussed these things also, bacteria and also bacteriocephalus. Other important points, note it down. We are the part of the end, I think. Important points, note it down, my dear. Alpha acti epithelium developed from a embryonic ectoderm. They may ask the question, olfactory epithelium is developed during the time of embryonic ectoderms. The correct sequence in spermatogenesis, they may ask the questions, spermatogenesis. Listen here, spermatogonia is mother cell, primary spermatocyte, secondary spermatocyte, spermatid, sperm. How many spermatid produced, my dear students? Ajay? Jaswant, how many spermatid produced during the spermatogonia or spermatogenesis? There are? Anybody else? 4 spermatid. Don't forget, spermatid 4. Egg is only one is there, isn't it? Don't forget. And second, gastrula end stage is characterized by the formation of neural tube. Gastrula end stage is characterized by the formation of neural tube. Okay, secondary oocytes is formed before fertilization. These are the important points. Egg is liberated from ovary in secondary oocyte stage. Note it down. This is very, very important. Okay, egg is liberated in the form of secondary oocyte stage. Primary oocyte stage also present inside only. Okay, when its secondary oocyte formation takes place, then only they can move from the fallopian tube to uterus, endometrium. Cells of lytics occurs in testes. Lytics. Site of fertilization in mammals is fallopian tube. Serotolic cells occurs in human testes. Serotolic cells. What is the main function of serotolic cells? They provide nutrients. Breaking of acrosome membrane is agglutination. Breaking of acrosome membrane is agglutination. The correct sequence of egg layers from outside to inside: proliferase, radiata, pellucida. P or P. Proliferase, radiata, pellucida. Third phase of embryo development is gastrulation. Okay, the correct sequence of the development is, which one you know? Fertilization, zygote, cleavage, morula. Fertilization, zygote, cleavage, morula. Already we discussed these things in the fertilization process, isn't it? Okay, we'll pass on to the next one. Any doubt in this area? And also morula and blastula and also gastrula. So here, C, what is that? Yes. So this is very, very important. Fertilization, zygote, cleavage, morula, blastula, and gastrula. MBC, MBC, note it down. Okay, you have to form as a mnemonic form as FZC, MBG, sorry. FZC, MBC, fertilization, zygote, cleavage, morula, blastula, and gastrula. Like that, you have to prepare. And then growth and maturation of the Graafian follicle is controlled by which hormone? FSH and LH. Growth and maturation of the Graafian follicle is controlled by follicle stimulating hormone as well as luteinizing hormone. After ovulation, Graafian follicles forms corpus luteum. Already we discussed. Corpus follicle, Graafian follicles forms corpus luteum. Fallopian tube is a part of oviduct. Okay, polar body developed during oogenesis. Oogenesis, what happened? There are two polar bodies also formed. Primary polar bodies and secondary polar bodies also formed. They are all are haploid in nature. Okay, first or free milk is called as colostrum. Colostrum, already I told you. The first mother milk is called as colostrum, which contain immunoglobulin A is very high concentrated, isn't it? Fixing up of blastocyst in the wall uterus is called as implantation. So many times we talked, we discussed, isn't it? Blastocyst, which one? Fixing up of blastocyst. in the wall of uterus, otherwise called as endometrium. That is called as implantation. Cleavage is repeated mitotic divisions of zygote. Cleavage, the cleavage is occurs due to the which type of division? No, mitotic division. Don't forget it here. It will not enter meiotic divisions. Here the mitotic, because it's also one of the somatic cells. Body cell division takes place within the egg. Okay, zygote. Function of serotolic cell is to nourish sperms, already discussed. Provide nutrient substances. Okay, number of sperm formed from 4 spermatocytes is 16. 4 into 4 multiplication here is the 4 forms spermatocytes gives 4 into 4, 16 sperms. Germ cells in female gonads and male gonads begins undergoing meiosis simultaneously. The ratio of ova and sperm produce are 1 egg and 1 4 sperm. Already told you. 1 egg, 4 sperm. This is called as oogenesis. This one is called as spermatogenesis. In anaphase, one of the cell divisions or oogonia arrested. Okay, anaphase 1, the oogenesis arrested. Placenta secrete estrogen. This is also another important point. Placenta secrete estrogens. Parturition, nothing but the delivery of baby from uterus. Gestation during, what is that? See that? During duration of the between the pregnancy and birth is called as gestation. Ovulation, release of egg from the Croftian follicles. This is called as ovulation. These are all are very, very important, my dear students. Don't forget, these are all very, very important points in examination point of view. Okay, so go to the next one. An implantation, already we discussed. Attachment of the zygote to the endometrium. Conception, stoppage of ovulation and menstruation is called as conception. Conception is nothing but stoppage of the ovulation and menstruations. The growth phase is the longest phase during the male gametogenesis. Sometimes they may ask the questions. Male gametogenesis, which phase is very longest phase? Growth phase. But human eugenesis, maturation phase is the longest. Growth phase, longest in gametogenesis. Human oogenesis, maturation phase is longest. Here the maturation phase is longest. Here, which one growth phase is the longest. The acrosome of sperm are produced by Golgi bodies. Note it down. This is very, very important one. The acrosomes of the sperm or head, protrude head-like structure is called as acrosome. Otherwise called as needle-like structure, a cone-like structure that is produced by the Golgi bodies. The smallest sperm is of crocodile and its size is 0.02 mm. The size of the sperm, very small one is which one? Crocodile. And the largest sperm is Discoclosus, 2 millimeter. 2 millimeter, 0.02 here, 2 millimeter. The 10 times more than the crocodile. Okay, this is very, very important, my dear students. 74 days or the required to the complete the cycle of spermatogenesis in human beings. Sometimes they may ask the question, the well-developed sperm, how many days it will take? That is 74 days. In 1 ml of semen contain how many sperms? See here, 20 to 120 millions of sperms are present in the human being. Deficiency in the number of sperm result of infertility, which is known as the oligospermia. Okay, infertility, the technical scientific name they have given as a oligospermia. Okay, absence of sperm in the semen is known as azoospermia. Note it down. These are all our clinical symptoms. Absence of sperm is semen is known as azoospermia. deficiency of number of sperm result infertility, which is known as the oligospermia. Number of sperm here, absence of sperm. Know the difference between these two. Largest egg is of ostrich, 60 centimeter long with its cells. Okay, note it down. 60 centimeter. Note it down. This is very, very important one, my dear students. Okay, go to the next one. Although normal number of sperm are present in the semen, but if there are completely non-motile in nature, this condition is known as necrospermia. Otherwise called as necrospermia. Suppose completely non-motile nature mean that condition is called as necrospermia. Do you know how many percentage of sperm in motile nature? How many percentage of the sperm is non-motile nature? My dear students. Ajay, you know the answer, my dear.
Sir, no, sir.
What about Jaswant?
Sir, no, sir.
Okay, already we discussed 100%, isn't it? 100% divided into two parts. Already told you. 60%, 40%. 40% is highly motile nature. Note it down, my dear students. Don't forget 40%. Okay. 60% will be normal and 40% will be very highly motile nature. Okay. That is, note it down. Here, non-motile, completely non-motile. What is the condition is called as completely 100% non-motile nature. How many percentage motile nature, my dear students? Both of you.
100%.
How much percentage?
100%.
No.
Sir, 0% the hotel, sir. Here.
No, Kana. What I am asking, normally the well health human being containing how many percentage of motile, vigorous motile? I am asking.
Sir, 40%, sir. Highly hotel.
Yes, very good. What about Jaswant?
Yes, sir, 40%. Sir.
Don't forget, 40% is correct. Okay, 60% is normal one, 40% vigorous nature. But in this category, what happened? I know 0%, 100% motile means what is that? Highly, isn't it? 0% motile, that is called as non-motile nature. That is necrospermia. Okay. The smallest egg in bed of hummingbird. Already you know which one is the, and backward flying bird is called as hummingbird. Don't forget it. Okay. Okay, my dear students. Path of sperm in mammals. Ajay. Hello. Ajay.
Sir, yes, sir.
Don't forget this line. What I am talking. Okay.
Yes,
So
sir.
this portion don't forget because it is very, very essential. Jaswant also do follow me. See that.
Yes, sir.
Yes, the path of sperm in mammals. How it will be entered? First, see here seminiferal tubules first. Tube first. Ready testes. Vasa efferentia. Efferent means outlet. Okay. Epididymis. Vasa difference. Urinogenital chamber and urethra. So many times they were asked the question in this part, my dear students. Path of sperm in mammals, seminiferal tubules, retidestes, and vasa efferentia. Then epididymis, vasa differentius, and urinogenital chamber and urethra. This is the path. Don't forget. S, R, V, E, V, U, U. See here. SRV, EVU, U. Okay. EVUU. SRV, EVUU. Don't forget. SRV, EDUU. Otherwise called as SRV, EVU squared. Just whatever it may be. Okay, don't forget. Already we discussed tubectomy. A method of population control is performed on females only. This is called as tubectomy. Tube, fallopian tube, female. Fallopian tube, female. Okay. And also Sogili, an oral contraceptive for female was developed by Central Drug Research Institute, Lucknow, Madhya Pradesh. This is also very, very important question. 1 and 2, 3. These are the 3 questions. Don't forget. And IVF involved transfer of into the fallopian tube of zygote or embryo up to 8 cell stage. Note it down. This is also very important. IVF. Okay, transfer into fallopian tube, zygote 2 or embryo up to 8 cell stage only they can used. MTP, okay, is considered as safe up to 12 weeks of pregnancy. Okay, I will tell you what is meant by MTP. GIFT is transfer zygote into fallopian tube. Okay, chemical spermicidal jelly and foam chemicals is another one. IUD, it is nothing but intrauterine devices, copper T and also loops. Barrier, just you noted on here, safety measures, sterilization, tubectomy and vasectomy. These are the important points. So another one is also already we discussed. This one is terms and explanations. Amniocentesis. Here you will not see clearly, mean just please refer the book. Amniocentesis, a fetal sex determination test based on the chromosomal pattern in the amniotic fluid surrounding the developing embryo. This is the correct explanation of this one. Here note it down. Sex determination 1 according to the chromosomal pattern. How? Amniotic fluid. Where it present? Amniotic cavity. Okay, surrounding the embryo. Lactation amnesia, lactational amnesia, absence of menstruation during extensive breastfeeding, mammary gland feedings. Lactation amnesia is nothing but the disease. Okay, sterilization, already in our sterilization, nothing but the surgical method to prevent pregnancy, tubectomy in female and vasectomy in male, vasectomy. Okay, artificial insemination, introduction of semen artificial into the female reproductive tract, insemination, artificial. You know very about the hybridization is also done by maximum animals. You know about Jersey and also Sindhu. They both are what happened. You know, the Jersey cow or buffalo present in the north side, Gujarat or somewhere else like that. You know, they collect the sperm, and in Tamil Nadu, take an example. Some of the female here, you know, the transport from that place to here. That is what is artificial insemination. Okay, so the sexually transmitted disease, the disease is infected which are transmitted through the sexual intercourse only. Reproductive health, physical, emotional, behavior and well-being is called as reproductive health. Medical transmission of pregnancy, MIP. This is called as medial termination of, medial termination of the pregnancy. Okay, international of volunteer termination of pregnancy before full term or induced abortion. In vitro fertilization, the fertilization outside the body, in almost similar condition as the in the body. In vitro means outside. In vivo, inside. Fusion of gamete within the female. WHO, World Health Organization. Where it located? Where it located, my dear students? Who?
Sir, Geneva, sir. Switzerland.
Very good. Who? Geneva. And then RCH, Reproductive and Child Health Care Programs. RCH, sometimes they may ask the question. MMR, sometimes they ask the MMR. Maternal mortality rate. IMR, infant mortality rate. IUD, intrauterine devices. This is 3 is very, very important. MTP, just we studied it. Medical termination of pregnancy, MTP. STD, sexually transmitted diseases. VD, venereal diseases. This is very, very important. The venereal diseases is called as syphilis. RTI, reproductive tract infections. Reproductive tract infections. PID, pelvic inflammatory diseases. ART, what is meant by ART? Associated reproductive technology. Assisted reproductive technology. IVF, in vitro fertilization or in vivo fertilization. This one is called as ET, embryo transfer. Okay, this part is very, very important, my dear students. After 23, just note it down, very, very important. ZIFT, without these questions, there is no need questions. ZIFT, zygote intrafallopian transfer. Zygote, Z denoted as zygote. IUT, intrauterine transfer. Here, zygote. Zygote is nothing but zygote intrafallopian transfer. Note it down. Okay. IUT, intrauterine transfer. GIFT, gamete intrafallopian transfer. These are same. GIFT and GIFT same. The first word only changed. Here the zygote, here the gamete. Here the zygote, here the gamete. Don't forget, my dear students. GIFT, that is called as zygote intrafallopian transfer. GIFT, gamete intrafallopian transfer. ICSI, intracytoplasmic sperm injection. Okay, this is another one. AI, artificial insemination. Next one, IUI, intrauterine insemination. And then last one is called as Central Drug Research Institute, Lucknow. So these are the important points. How many is that? 29 is here must. Okay, study, don't lift it. Okay, this is very, very important, my dear students, because this question, this tabulation is, go through it thoroughly and pick up your own point. Which one is very easily it comes out. Like that, you have to practice it, then only better for your future. Okay. So good luck, my dear students. That's all about these lessons. Human reproduction and health lesson is completed thoroughly. Suppose if you have any difficulties, that the two things is you must note it down. What is that? You know, genital organ of female, genital organ of the male. Just to go through the book, you will get the idea, isn't it? Okay. Okay. I will give you some small intro only for the next lesson. Then we'll pass on to the another one. So this lesson is finished. We'll pass on to the next one. Is called as, what is that? See that, principles of inheritance. What is my inheritance, my dear students? How it will be inherited from the one generation to another generation? Principles of inheritance and variations. Inheritance is nothing but what is that? Presence of DNA or gene. They can transfer from one person to another person, or one generation to another generation. That process is called as inheritations. What is by variation? When it happens, variation occurs during the mutation. The mutation may occur due to some three factors: physical, chemical, and biological factors, isn't it? So this is also one of the mutations. Just already saw the Jurassic Park, isn't it? So this is also one of the, just for examples. Don't think this is not a lie like that, you know. This is the one. Now the examples I have taken for your visible aspects. But first of all, we should know what are the technical term is the basic term is known. First one is gene, alleles, autosomes, factors, and then linkage, Punnett square, incomplete dominant, mutation, mutagen, point mutation, hemophilia, Klinefelter syndrome, okay, phenylketonuria, and sickle cell anemia, pedigree analysis, and homozygous, heterozygous, okay, and genotype, phenotype, F1 generation, F2 generation, and genetics. So these are the, what is the basic terms you must note it down, then only we can enter the things. Before we talk, okay, what is my gene? What is the main use of genes? You look like your father. Why you look like your father or mother like that? What are the general characteristics of your father? What is your special characteristics of your family or your generation? Like that they may ask the questions. Okay. Excuse me. So these things we will see the next classes. Okay, my dear students. So any doubt, my dear students?
No, sir. No doubt, sir.
No, sir.
Okay, just to go through it once again, I repeat for you. Please go through it. We'll get the answer. Okay. If you have any doubt, you can ask me. Okay. Thank you, my dear students. Bye. Take care.
Thank you,
Thank you,
sir.
sir.
Thank you, ma'am. Thank you.
Thank you, sir.
Thank you, ma'am.